This episode of Cell and Gene the podcast is brought to you in partnership with Thermo Fisher Scientific. Thermo Fisher's cell therapy processing instruments are designed to help customers transition from process development to commercial manufacturing, utilized as standalone devices or integrated as part of a closed modular process. Thermo Fisher Scientific recommends Gibco CTS DynaSelect Magnetic Separation System, which is a next-gen cell isolation and activation instrument. Gibco CTS Xenon Electroporation System allows customers full control to optimize for a variety of cell types and payloads. And Gibco CTS Rotea Counterflow Centrifugation System is a closed cell processing system supporting a broad range of protocols for cell separation, washing, and concentration. Customers can rely on and streamline their drug development process with Applied Biosystems QualTrack qPCR and dPCR quality control tools for robust and reliable genetic analysis across various phases of drug development, supported by relevant, compliant documentation. Hello, listeners, and welcome to this episode of Cell and Gene the podcast. I'm your host, Erin Harris, and my guest for today's episode is Vittoria Biotherapeutics CEO, Nick Siciliano. Vittoria Bio is based in Philadelphia and is focused on expanding the applications for CAR-based therapeutics by employing novel cell engineering and gene editing technologies. And we'll get to all that in just a second. But first, Nick, welcome to Cell and Gene the podcast. Thanks for being here.
Thanks, Erin. Thank you for having me.
Absolutely. So let's jump right in. You are the CEO of Vittoria Biotherapeutics. And like we said, it's a gene edited cell therapy company. You have a novel platform technology exclusively licensed from the University of Pennsylvania. Now that's a really broad description of what your company is and what you do. And so I was hoping you could break it down for us exactly what Vittoria is working on, why it was spun out of Penn, and kind of what you're doing to get to the path of unmet need.
No, that's great. And obviously a lot to cover there. So, I'll start with, obviously we're, really passionate about the promise of cell therapies. I mean, we believe fundamentally that, this technology, could truly be, the future of medicine from a modality standpoint. And I also think that, we're fortunate enough to be working here in Philadelphia, where, there's a number of pioneers at the University of Pennsylvania whose experience we can lean on and benefit from, as it relates to, what we see with the first generation of cell therapies. And, I think there's been a lot learned, and I think there's a lot, that can be improved on. And really that's the genesis of Vittoria and kind of our vision, is to work on the next generation of cell therapies and to leverage all of the data and experience, that the industry, has gained here from the first generation, specifically, again, the pioneers that we're fortunate enough to partner with at the University of Pennsylvania. And, how can we address the specific shortcomings, and those range from really just, limited approved indications. Right now, we only have approved CAR T therapies for B-cell malignancies. Efficacy, there's always room for improvement there. Typically, 30 to 40% of patients that are treated today, achieve a complete response with long-term durability. I think obviously, we all aspire to see that number go up dramatically. There are some, general safety and toxicity issues with conventional cell therapies that we also believe can be improved on. And then, of course, there's the manufacturing and logistical challenges that just are inherent to some degree, with a cell therapy approach. But again, I think there's a lot of work being done in that space to improve on that. And, we're focused on that as well.
Good. Okay. All right. So I want to talk a little bit about the technology Vittoria is attempting to commercialize here. So the technology itself was developed by Dr. Marco Ruella. He's the assistant professor of medicine in the Perelman School of Medicine and a member of the Penn Bioengineering Graduate Group, who is the company's scientific founder. And the Perelman School of Medicine, I'm very thankful we have friends of Cell and Gene in the podcast that we've worked with, who are, like you said, pioneers of the entire CAR T cell therapy space that we've worked with. And I wanted to, though, talk to you a little bit about the technology you're attempting to commercialize here. What is it? Talk us through it.
Sure. So again, kind of, aligned with our vision of generating, the kind of the next generation of cell therapies and improving upon, some of the drawbacks with the conventional cell therapies. One of the things we've looked at is, okay, what are the specific drawbacks? And then, what tools do we have to be able to address them? So one of the biggest advances over the last decade, since the team at Penn first started dosing patients with what is now Kymriah, has been, the development of gene editing technologies. And one of the things that Marco was focused on is, how can we leverage, gene editing to solve for some of the issues that we've identified as it relates to potency of cell therapies? How can we make them more potent? How can we use them, leverage gene editing to treat more indications? So that's kind of where, a lot of his research was based. He was recruited to Penn to work with Dr. Carl June and team, and actually started at Penn working in a laboratory that was focused on correlative studies. So they were actually investigating patient responses to, essentially what is now Kymriah, and looking to see, was there interesting patient data or anything they could glean from patients who were achieving complete responses versus patients that, were only achieving partial responses and not responding. And he identified a number of attributes in the cells in patients that achieve complete responses. And is there a way that, knowing what those correlates are, knowing what those specific attributes are, could we engineer a cell therapy that would be better at achieving that in a broader population? So, Marco, and, as his career's evolved at Penn, he's really become a rising star in this space, decided to, look at how can I leverage gene editing on solving for some of the challenges with broad efficacy with cell therapies and also expanding indications. So the technology he's developed is essentially utilizing gene editing to delete an important signaling pathway in T cells that's known to be an inhibitory pathway as it relates to effector function. And that pathway is the CD5 signaling pathway. So by deleting it, utilizing gene editing, we basically get a gain of function effect. And we get a CAR T cell that's much more prolific from an effector response standpoint, and also solves for, some of the observations that he had as it relates to what are important clinical correlates, with CAR T cell therapies. And, some of those are rapid expansion of the CAR T population, early after infusion of the patient. So you get this, obviously larger effector population of CAR T cells to be able to treat and, kill cancer cells. So that's something that we're able to achieve with deleting CD5. And we call the platform Senza5, which essentially means without 5. But the other thing that he identified is, this is very broadly applicable. So we investigated it in a broad set of liquid tumor and solid tumor models to really show that you see this enhanced efficacy across the board, and it's a generalized effect. And in addition to the enhanced early proliferative properties, we also see this longer term durability and resistance to exhaustion, which is another challenge, in patients that aren't achieving long-term responses, aren't achieving complete responses, is this concept of the CAR T cells eventually getting exhausted and failing to be cytotoxic to the tumor cells. And basically you get, the tumor escape and essentially winning. So, this our Senza5 platform allows us to really achieve the best of both worlds. We get this early proliferative effect. So you get a bigger population of effector cells, and then all of them are resistant to exhaustion. So you get this longer term durability. And again, we've seen it in every animal model we tried it in across liquid and solid tumors. So we think it's a great platform technology that will be plug and play for, different CAR Ts, not only for liquid tumors, which our lead program is focused on, but also for really what has become the holy grail for CAR T therapies and solid tumors.
Solid tumor, sure. Okay, thank you for that. That's really helpful for our listeners to better understand exactly what it is you're working on. I want to talk a little bit about from a broad cell therapy perspective. Now, you referenced, of course, broad applicability, gene editing, all makes perfect sense. But when I was researching your company, I learned that Vittoria, its technology and clinical programs are designed to address the current gaps in cell therapies. And that's what I wanted to talk to you about broadly. What are those really large gaps that exist, and how are you and your team working to fill those gaps? And even you as CEO, what's your role in all of this?
No, that's a great question. I'll start with the last one. Is my role as kind of CEO. I think that there's a tremendous responsibility on all of the companies in this space right now, and I take that responsibility personally. To make sure that we're successful, because our success is really going to determine, does this modality become mainstream? Can we treat more chronic diseases with this modality? Because again, I think it's an important transformative one with curative potential. Can we make it, easier to manufacture? Can we make it more accessible to broader patient populations? And I think that's a responsibility that we all bear, and that's something that, again, we're hyper focused on here at Vittoria, to making sure that, we solve for some of those issues so this field as a whole can, advance and, again, realize its full potential. As it relates to specifically what are some of the drawbacks, again, potency and durability of the product are a big one, especially when you start to think about solid tumors, because solid tumors, one of the things you're up against is this tumor microenvironment. And the tumor microenvironment has basically evolved to become immunosuppressive and to basically keep the immune system from clearing, malignant or tumor cells. So, you've got this inherently immunosuppressive environment that's been, that basically has been programmed to diminish, T cell effector functions, et cetera. How do you overcome that? So, you really got to create a T cell that's not going to be sensitized to that, or that's going to have resistance to some of the exhaustive properties that it's going to be up against in that tumor microenvironment. And again, that's something that, we've been focused on with our platform technology, and we've shown in a number of models that our platform technology, not only kind of broadly do we see that enhanced cytotoxicity in these solid tumor models, but when we look at it mechanistically and we look at it on a molecular level, we can see the differentiation between a Senza 5 CAR T cell and a conventional CAR T cell, and that it's not getting exhausted. It's not showing phenotype or phenotypic exhaustive properties or markers, et cetera. And, we're starting to we've dialed into a number of molecular mechanisms as to why. So, we think that that's really obviously important to solve for, and again, all kind of comes back to potency, durability of, of response, et cetera. As far as indications go, I think that, gene editing also gives us, and our platform technology in and of itself gives us the ability to expand indications. I think a great example of that is our lead program, Viper 101 for T cell lymphoma, where we are targeting a T cell with an engineered T cell or CAR T cell. So there's some inherent challenges there. And one of those is, if you're targeting T cells, you're typically targeting a pan T cell marker that's also going to be expressed on the engineered cell therapy. So if you simply target a pan T cell marker with a CAR T cell, the CAR T cells can attack each other. And that's called fratricide. So that's a fundamental drawback that with our lead program, we're able to leverage gene editing and specifically editing CD5 to avoid that effect. So there's an example of, again, how we can, use our platform technology to kind of expand and treat indications where there's a huge unmet medical need, so that's exciting for us as well. And then on the manufacturing side, we've really synergized that platform with a shorter autologous manufacturing protocol. And it's a novel one that we've developed here at the company that's only 5 days. So 5 days from start to finish, you get a gene edited CAR T product. And what's interesting is that comes along with a number of advantages from a manufacturing efficiency standpoint, from a cost standpoint, from a bill of materials standpoint. There's a lot of efficiencies that can help realize. But interestingly and importantly, it also confers a functional advantage to the drug product. So, there's a number of studies that now show, and a lot of them actually were done here at Penn and published out of Penn, that cells that experience less time ex vivo in culture and, are engineered with these shorter autologous manufacturing times do better in vivo.
Sure.
And to some extent, that's intuitive, right? These cells, don't want to be in an ex vivo environment. They're used to being kind of in the body, et cetera. So the less time they're in, I'll call it an artificial environment, you can see how the better off, they'll be phenotypically. And that's been shown to be true. So we basically combined the Senza 5 platform technology with a 5-day manufacturing process. And again, we show in the models that we've investigated in that additional or synergistic effect of the functional advantage of what we consider to be our commercial embodiment of the technology.
That's incredible. I do have a quick question about the 5-day manufacturing process. Are you doing that in-house? Have you partnered with another company? How are you doing that exceptional turnaround?
Great question. So now we've partnered with a CDMO, and we've we developed the manufacturing protocol. So the manufacturing protocol is the companies, but we partnered with the CDMO using their GMP suite and operators in order to manufacture for at least our initial phase 1 study.
Sure. A local company? Yes. Local to Philadelphia?
Yep.
Nice. Okay.
Wuji Aptech.
Sure. Yeah. In the Navy Yard?
Yep.
Yep. Excellent. Okay. Great company. All right. So thank you for that detail. That's really helpful. So we're in Q4 of 2023. Talk to us about what does 2024 look like for Vittoria?
It's really, we're super excited about 2024. It's going to be a milestone year for us. So we've recently submitted our IND and expect to be dosing patients starting in early 2024. So obviously the most significant value inflection milestone for the company kind of near term here, both from the value that we can create for prospective patients, as well as, of course, the value we can create for our shareholders. So we're really excited, expect to be dosing patients kind of in the first half, if not Q1 of 2024.
Oh, that's excellent. So then I hope we can reconnect maybe midway through next year. You can bring us up to speed on how that's going, and I'm certain progressing really nicely.
That would be terrific.
All right, listeners, that wraps up this episode of Cell and Gene, the podcast, featuring Vittoria Biotherapeutics CEO, Nick Siciliano. Thanks for tuning in, and we'll talk to you soon.